Welcome, everybody. Today, we have the pleasure to have with us Aina Oliver-Caldés from the Hospital Clínic de Barcelona, University of Barcelona in Spain. And we will talk about a recent paper in Lancet Oncology entitled Fractionated Initial Infusion and Booster Dose of ARI-0002H, a humanized BCMA-targeted CAR T-cell therapy for patients with relapsed or refractory multiple myeloma. A CAR T BCMA HB01, a single-arm, multicenter academic pilot study. First of all, thank you for accepting our invitation. How are you today?
Very good. Thank you, Carmelo, for having me. It is an honor.
And so let's go to the nitty-gritty. Can you explain the rationale behind the use of CAR T cells in myeloma?
Yeah, sure. So in multiple myeloma, we have on one hand the fact that now we have several treatments for these patients that we can use nowadays. But at a certain point, when we already have used proteasome inhibitors, immunomodulating agents, anti-CD38 antibodies, in this, let's say, multi-drug resistant setting, it has been shown by some studies, such as the locomotion, for example, that there is not a clear standard of care therapy for these patients, and the combinations that we use only lead to median PFS that are shorter than 5 months. And then we have, on the other hand, this nice fact that is that hematologic cells, and specifically myeloma plasma cells, express some antigens on their surface, such as BCMA, that are rarely expressed in healthy tissues. And this makes them a great target for immunotherapy. And I think that the combination of both, the need to find new treatments and the fact that they are targetable by immunotherapy, has led to a big success of CAR Ts in multiple myeloma.
Thank you. So, as you said, we are talking about CAR T cells in general, but there are various types of CAR T. So, can you tell us which kind of construct did you use in your study and why, and if there are any advantages that you have envisioned in this?
Yes, so we decided, so RE2H is a 4-1BB second generation CAR that is targeting BCMA, and it is lentivirally transduced on autologous T cells. And the distinctive feature that it has is that the scFv has been humanized with the aim to diminish xenoreactivity and try to increase the persistence of the CAR.
Okay.
That would be, I guess, the most distinctive feature.
Okay, thank you. And could you give us instead now an overview of the study design, for instance, why you use the fractionated dose and the characteristic of the patient enrolled in this study?
Yeah, sure. So this pilot trial was designed to include relapsed refractory multiple myeloma patients. Inclusion criteria, amongst others, were age between 18 and 75. They had to have received 2 prior lines of therapy at least, including proteasome inhibitors, immunomodulating agents, and anti-CD38 antibodies. And they had to be refractory to the last line. So patients undergo screening, apheresis, and while the product is being manufactured, they are allowed to receive bridging therapy. And once the product and the patient are ready, we start lymphodepletion with fludarabine and cyclophosphamide from days -6 to -4 from infusion. So at this point, the first dose of 3 million CAS per kilo is given in a fractionated way. So this means that it is split into 3 fractions of 10, 30, and 60% of the total dose. So we start on day 0 with the first fraction of 10% of the dose. And if no side effects come up, then we continue to the following fraction and so on, always keeping 24 hours between fractions. But if at some point a side effect is observed, then the following fraction can be postponed until resolution of the symptoms. That would be like a very distinctive feature. And the other thing that is peculiar in this trial is that there is a second infusion of CARs, again with 3 million CARs per kilo, but only in one fraction. And this second infusion is planned after 3 months of the first infusion in patients that have presented at least some degree of response to the first one and that did not present like limiting side effects. And for this second dose or booster dose, that's how we call it, we only give lymphodepletion to patients who do not have persistence of the CAR in the peripheral blood. Otherwise, they go straight to receiving the dose. So the aim of this booster dose was to try to give the patient some fresh CARs, to try to lengthen and deepen responses, because we believe that these CARs that we give in the booster dose are potentially less exhausted, to say it in some way, right? So in the paper, as you said, we presented the results of the first cohort with 30 patients. The median age was 61, and almost half of them had an ISS of 3. 1/3 of the patients presented with high-risk cytogenetics, and 20% had extramedullary plasmacytomas at baseline. And then the median previous lines was 3.5, with 67% of triple refractory and 23% of penta-refractory.
Wow, so that was like definitely a very challenging setting. So tell us, how did this patient do? Guide us through your result as to overall response rate, complete response rate, MRD negativity. I've seen you performed this analysis and also some imaging studies, right, with the PET-CT scan.
Yeah, exactly. So in terms of response, all patients responded within the first 3 months, which was the main efficacy endpoint of the study, with 50% of complete responses. But since in some patients receiving CARs, we've seen how responses deepen over time, we ended up having 67% of complete responses or better, 27% of VGPR, and 7% of PR. And regarding MRD, in this trial, MRD was measured with next generation flow at a sensitivity of 10 to the -6 in Bruno Paiva's lab. So all the samples were centralized. And on day 100, we had 92% of the evaluable samples that were negative. Then you mentioned imaging response, yeah. So we evaluated the response of the plasmacytomas with a PET-CT on day 100 also, and we saw a metabolic response in 13 of 14 patients that presented plasmacytomas at baseline. And then in terms of survival in general, with a median follow-up of 18 months, median PFS was 14.5 months. And median duration of response and overall response were not reached.
Very nice results. And now let's go back at the infusion times. Were there any side effect, CRS, ICANS? Let us guide us through the safety and the side effect of this construct.
So in these first 30 patients, we had 80% of them presenting a CRS, but all of them were grades 1 or 2. We had no grade 3 or higher of CRS. And in fact, regarding what I mentioned about the fractionation of the first dose, it was decided that in 5 patients, the third fraction of 60% would not be administered due to CRS. Okay. And when we analyzed responses and PFS in these patients compared to the ones that received the full dose, we didn't see any differences. So this is probably because these patients experience a higher expansion in vivo of the CAR, and they probably don't need the full dose, to respond. And the signs that we see developing maybe persistent CRS or, that makes us think that we shouldn't give the third fraction, right, is related to this higher expansion in vivo.
Got it.
And then in terms of neurotoxicity, we didn't see any cases of ICANS.
Oh, nice.
And we haven't seen late neurologic events so far. And yeah.
Perfect. Then how long did the CAR T cell persist then in the blood? What was the median half-time in this patient? Was there any correlation between the persistence of CAR T cell and the response of this patient?
So median persistence of the CAR T in the peripheral blood was 5 months.
Okay.
And we didn't see a correlation with response. In fact, we have both. We have patients who, despite losing the CAR, maintain the response for a long time.
Mm-hmm.
And we also have patients that relapse with persistence of the CAR.
That's like so fascinating. Like we still have like so much to learn from.
Yeah, exactly.
Like, from
exactly. In fact, we are working
therapy.
on it. Yeah.
And how about the BCMA expression on the patient who relapsed? Did they lose it or not?
No. So in terms of BCMA, we didn't see any antigen negative relapses.
Okay.
With all patients presenting BCMA on the surface of the plasma cells at relapse. But since we didn't have bone marrow samples of all of the patients at relapse, we decided to check soluble BCMA levels, and they were all positive. So we didn't have any antigen negative escapes.
Okay. As you mentioned, some patients got the booster dose, right, at 3 months after the initial dose. Did they experience longer remissions?
Yeah, that is a tricky question. So the booster dose was given to 24 of the 30 patients. And of these 24, 14 already had a stringent complete response at the time of receiving the booster dose. So of course they could not improve the response. And we don't have a way to know if it lasted longer. Okay. And from the other 10 patients, we have 6 patients who maintained the response. And this was a very good partial response. And then we have 4 patients of these 10 patients who improved their response, 2 from partial to complete response, and 2 from very good to complete response. Okay, so since there is not like a comparison randomized group, we cannot say that these improvements are only attributable to the booster dose. But still, taking into account the feasibility of manufacturing this booster dose and the lack of side effects observed, which make us even consider an at-home management. And then we have this potential benefit in terms of efficacy. So we believe that it would be nice to have this booster dose further investigated in other studies.
Okay, so I think time will tell us, right?
Yeah, exactly. Time will tell, yeah.
So what do you see now that we are talking about time in the future for this CAR T product? Because it was also nice, the pharmacoeconomic reasoning that you have discussed about in the last part of your paper, with emphasizing that nowadays in Spain there's no commercial CAR T product available for myeloma patient, and you gave this academic option with a manufacturing point of care. So do you think this will be able to supply the demand of your country? Like, and if so, do you think this is translatable or also to other countries? What do you think about this?
Well, so yeah, I hope it is like that. We need to go like step by step, you know. So now we can already use RE2H under a compassionate use program that we have here in the country, in which apheresis are received at our institution, hospital clinic, where the manufacturing takes place. And then the patients are treated locally in the centers that have participated in the clinical trial. So patients don't have to move to Barcelona.
Okay.
They only send the apheresis and we send back the final products. Okay. Now we are pursuing the approval of RE2H under a hospital exemption rule of the Spanish Medicines Agency. And with this, we would have like the support of the Ministry of Health in terms of economics. Okay.
Okay.
So it could be administered to any patient around Spain. Because now the competency of the price of the payment depends on the region of Spain.
Okay.
It's not something centralized to the whole country. And we really hope that the idea is that many more centers start manufacturing in the near future. So yeah. Talking about Europe, I don't know. We want to get first to this the system in the country and see if we can then go to the European agency.
I see that, but I thought it was like brilliant because of course that's like a very nice a prototypic example, I will say, of how academia can also like be, if you wish, competitive and also provide with an option that is at least as good as the one that we have commercially available. So really congratulations on the paper and also on this specific aspect, which is not to be underestimated. So thank you for this, Aina.
Thank you, and I think it is very important to acknowledge everyone participating on this, because this is a very big project, and making it work in a public institution, in academia, it's very hard. And despite that, we were able to write the paper. There has been a lot of people working on this. So I mean, they deserve this recognition, I think. Yeah.
I agree. We can't emphasize how important is the team effort, as
Exactly.
you mentioned, in this kind of like effort. So please, like, absolutely congratulations to everybody, all the team involved, and again, thank you for being part of the Trainee Pearls.